We are now at the point where we need to really start to explore the structure of the ribosome because we now have a really good understanding of the overall process. We have a good understanding of the molecules involved, the tRNAs, the aminoacyl tRNAs, the aminoacyl tRNA synthetases, and we have a good grasp of what those aminoacyl tRNA synthetases need to look like to be able to do the chemistry that they do, right? To be able to select the right amino acid and pair it with the tRNA that presents the correct anticodon. So the ribosome, as we said in a previous video, is the large RNA protein complex that catalyzes the reaction. Okay, and we mentioned before that it contains two subunits, the large subunit and the small subunit. So the large subunit we call the 50S, and the small unit the 30S. And remember, these are relative to E. coli. Okay, these Svedberg units are not additive because collectively, when we put them together, it's 70S. So remember, the Svedberg unit just tells us that 70S means that the complex is larger than the individual large or small subunits. Okay. We also mentioned that in addition to the 3 chains of RNA, there are proteins involved in the ribosome, right? So the large subunit has the 23S and the 5S RNA in addition to 34 proteins, whereas the small subunit has 21 proteins and just 1 chain of RNA. And these colors here correspond to the colors in the images. So in the small subunit, we see blue proteins here, and the green is the 16S ribosomal RNA. In the large subunit, we have the 23S, and if you look really closely here, you can pick out, oops, sorry, not there. If you look really closely here, you can pick out the 5S ribosomal RNA. And then the proteins are here. And when we put these together in the full 70S ribosome, the really interesting thing is that the interface between the two subunits is all RNA. There's no proteins at the interface. The proteins very much decorate the periphery, right? So if you look at all these proteins, they really are on the outside. So they're there for structural purposes, and in some ways, actually many ways, they're there for regulatory purposes. But they really don't play a role in catalysis, okay? So these are all kind of things that we've already talked about, is that it's primarily an RNA molecule, okay? They are ribozymes, so 2/3 of the mass is RNA, okay? These RNA molecules fold into complex structures with lots of these little short duplex regions. So like those are the stem loops that we talked about with the tRNAs. And then the ribosomal RNA is genuinely the catalyst for protein synthesis. The proteins really don't do a lot. They're there for structural and regulatory reasons. Okay, looking closer at the active site where the action happens. So here we see that there are 3 distinct tRNA binding sites. There's the A site we see here. There's the P site and the E site. So the A site stands for the aminoacyl site. This is where the incoming aminoacyl tRNA binds. The P site stands for peptidyl transfer site. This is the location of the growing peptide chain. And then the E site is once the peptide chain has been transferred onto the new tRNA, the E site is where the unloaded, uncharged tRNA is, and it's able to exit. And you can see here that there's really like no structural bulk from the ribosome that's preventing the tRNA in the E site from exiting. Whereas if we look a little bit closer here, we can see that the tRNA in the A site and in the P site have some structural features of the ribosome on top of it, preventing them from coming in or going out. Okay, let's look at the interactions between the tRNA anticodons and the codons on the mRNA a little bit more closely. So the colors here correspond to the colors that we observe here. So this is our A site, this is our P site, and this is our E site. Okay. The one thing that I really want to point out here is that the strongest interaction, the strongest base pairing interaction between the codon mRNA and the anticodon on the tRNA is present in the A site. We see that all three nucleotides have some sort of a base pairing interaction. Okay? That's really only present at the A site. In the P site, we see at least one of the anticodons are now looped out, no longer making a hydrogen bond with that third codon. Okay? And in the E site, there's really no hydrogen bond interaction. There's just this one kind of pi stack there that loosely keeps it in place. So there's very, very little interaction at the E site and maximal interaction at the A site. And if you think about the role of each of these sites, I hope that makes sense. The A site is the initial site where the tRNA comes in. It's the site that has to make the maximal recognition between the codon and the anticodon. We don't want a mistake to be made there. So we want to make sure there's an interaction with all three nucleotides. Once that tRNA has moved to the P site, it doesn't really matter anymore. The decoding process has already happened, so it doesn't matter if we no longer are making maximal interactions. The decoding has already happened. So how does this happen? So let's look at a schematic here. So here is the overall ribosome. The mRNA we see here, it's bound to the small subunit. All of the chemistry happens at the large subunit. Okay, so the idea here is that the tRNA in the A site, the amino acyl tRNA, this is the incoming amino acyl tRNA. A single amino acid is present here, right? That can also be seen here, a single amino acid in the A site. The P site is where we have the growing peptide chain. And this right here is the polypeptide channel, so the growing peptide or protein can exit the ribosome as it gets longer and longer. And then here we see the E site where this tRNA is not charged at all. There's no longer an amino acid present on the 3' end. So the idea here is fairly straightforward, and it's really what we talked about before in the very first video. The N-terminus of the amino acyl tRNA in the A site is the nucleophile. This carbonyl in this ester linkage between the tRNA and the growing peptide chain is the electrophile. We get nucleophilic attack of this lone pair onto the electrophile, ultimately breaking this bond right here. And that results in this entire polypeptide chain moving over into the A site as we see here. So after this polypeptide or this peptide bond has been formed, the entire peptide chain is now present on the tRNA in the A site. And that's meaningful, right? Because if you think about what's happening here, the peptide channel is present and positioned in a way where the peptide chain needs to be in the P site. So if the peptide chain has moved over to the A site, right, this all has moved over here, then that creates a really big steric clash. It creates a lot of stress on the system. So that gives the ribosome a lot of initiative, a lot of reason to do the translocation process for the entire mRNA to move over one position, bring this tRNA with it, move this tRNA over to the E site, right? Because of this stress, this entire thing wants to move over to here. This happens, it's a process called translocation, and it relies on hydrolysis of GTP, okay? When translocation happens, this tRNA is ejected, the tRNA in the P site moves over, and the tRNA in the A site that has this big long polypeptide chain also moves over. Okay, and I realize there's a lot going on here, so let's try to reiterate that one more time. Upon GTP hydrolysis, this tRNA exits. This tRNA, which now is unloaded, moves over to the E site. It's now an uncharged tRNA. This tRNA that now has this amino acid and the rest of the amino acids moves over to the P site. It now contains the entire polypeptide chain on it. And that then frees up the A site for a new amino acyl tRNA to come in. Okay? And the entry of that new tRNA is also GTP dependent, and we'll look at that process here shortly. But first, go ahead and take a few minutes to work on the next question on your activity.